Hi student, this is Professor Harmony Kim. This video record is for the lecture about cell structure and function. So far we studied atom, and atom form chemical bond with other atom to form molecule. These molecules associate with other molecules to produce macromolecule, and macromolecule also associate with other macromolecule. It forms organelle. And these organelles compose cell. So today you will study the cell structure and function. Cell theory. All life is produced from cells and all life is derived from the division of cell. All cells arise from pre-existing cell. A cell does not come out of nothing. Cell always come out of pre-existing cells. This means that cells are the basic unit of life. I told you several times, basic unit of life is a cell. Basic unit of matter is atom. The fundamental unit of life is the cell. The smallest organisms are unicellular. In other words, single cell is entire its body. Cells are the functional unit of multicellular organism. Your body is composed of a gazillion number of cells. All cells share a common feature. All cells have a plasma membrane, also cytoplasm. Cytoplasm is a semi-fluid substance called cytosol. In other words, inside the cell there is a nucleus. Except nucleus, the rest part we call that cytoplasm. This cytoplasm is filled with semi-fluid substance. We call that cytosol. So cytoplasm is more spatial concept. The space outside the nucleus, inside the cell, we call that cytoplasm. Cytoplasm is filled with some substance. Those substances, cytosol. You can say cytosol and cytoplasm in a similar concept. DNA as hereditary blueprint. DNA is always localized in the cell, especially in the nucleus. RNA to copy the genetic blueprint and guide the construction of a protein. These features can be shared with any kind of cell. Let's look at this figure. Can you see this plasma membrane? It has a phospholipid bilayer. Throughout phospholipid bilayer, this blue chunk structure is protein. Proteins are embedded throughout phospholipid bilayer. This embedded protein play a role of communication portal. Therefore, these proteins regulate passage of certain molecules and ions. Phospholipid bilayer interspersed with cholesterol. This is cholesterol. Cholesterol is steroid lipid. Therefore, it has a hydrophobic property. That's the reason why cholesterol is embedded throughout this tail region of a phospholipid. Do you remember? Tail region is a hydrophobic property. Head region carries a hydrophilic property. Cholesterol is a hydrophobic molecule. Therefore, cholesterol resides in phospholipid tail region. This figure shows again plasma membrane. You can see phospholipid bilayer, and this purple color indicate transmembrane protein. So based on the phospholipid bilayer, the below part is inside the cell. That's why here's the cytoplasm. The above part from phospholipid bilayer is outside the cell. We call that extracellular matrix. And phospholipid bilayer, you can see cholesterol and then some other proteins also integrate throughout phospholipid bilayer. And you can see also this green hexagonal structure. This is carbohydrate. Therefore, carbohydrate bind to protein. We call that glycoprotein. Glycogen, glucose, these represent the carbohydrate portion. And protein. carbohydrate and protein bind together, we call entire structure as a glycoprotein. So you can see there are several glycoproteins located throughout phospholipid bilayer. Again, when you look at phospholipid bilayer, you can see carbohydrate side chain bind to protein, indicate glycoprotein. If we look at this cubic box, here's a hydrophilic region. The bottom part is a hydrophobic region. Again, hydrophilic region. In other words, here's plasma membrane. Inside the plasma membrane, in other words, the cytosol area is liquid state, therefore hydrophilic region. And this tail portion of phospholipid bilayer is hydrophobic region. And outside the cell is watery environment, therefore, again, hydrophilic region. Then carbohydrate face outside because carbohydrate has hydrophilic characteristic. This is entire cell in three-dimensional structure. Cytoplasm, all the fluid and structure inside the plasma membrane, but outside the nucleus. So you can see cytoplasm, so many organelles and molecules filled in cytoplasm. Cytoplasm is the site of most biochemical reactions take place to support life. Cytosol, fluid portion of cytoplasm. So when you look at this cytoplasm space, there's all space is filled with something, some molecule, macromolecule, and even some liquid state. Therefore, fluid portion of cytoplasm is called cytosol. Water, salt, organic molecules dissolve with fluid. DNA and RNA, we already studied about this. DNA located in every cell. There is no cell which does not have DNA. Hereditary blueprint. DNA hold gene segment. These genes store information. So DNA construct protein down the road and also produce new cells. RNA, chemically similar to DNA, but RNA just copy genetic information from DNA. RNA help construct protein. On ribosome, this is the site where protein synthesis takes place. This ribosome is composed of protein and RNA. So ribosome is a cellular workbench of specialized RNA. There are two basic types of cell we already studied, prokaryote and eukaryote. The main characteristic of a prokaryote is it lacks of a membrane-bound system. Eukaryote has membrane-bound system. So main criteria to differentiate between prokaryote and eukaryote is whether the cell has a membrane-bound system or not. Prokaryotic cells do not have membrane-bound system. Lack of membrane. Eukaryote has membrane-bound organelle. So prokaryote do not have a nuclei, but eukaryote has a nuclei. What does this mean? I will explain the concept of a nuclei a little bit later. The basic structural and functional unit of every organism is one of two types of cell, either prokaryote or eukaryotic. Only organisms of the domain, bacteria and archaea, consist of prokaryotic cells. Living organisms belong to bacteria and archaea do not have a membrane-bound system. Therefore, they belong to prokaryotic cells. Protists, fungi, animals, and plants, they all carry membrane-bound organelle. So, these living organisms has eukaryotic cells. The characteristic of a prokaryotic cell is unicellular. Single cell is entire its body. There is no nucleus. The name nucleus itself include having membrane, nuclear membrane. Since prokaryotic cells do not have a nuclear membrane, we call that prokaryotic cells do not have a nucleus. Without nucleus, how DNA present? DNA in an unbound region called nucleoid. There's a DNA macromolecule, but these DNA is exposed in the cytoplasm. So actually there's no cytoplasm and nucleus delimit. There's no nuclear membrane, therefore we cannot say this portion is nucleus, the rest portion is a cytoplasm. We cannot even differentiate that way because there's no nuclear membrane, therefore inside the cell is all same condition. Then DNA is just unbound and then clustered. We call that nucleoid. Most feature plasmid outside the nucleoid. So there's one E. coli and then DNA is like this clustered. And then there's some small circular ring structure. We call that plasmid. This is also very short DNA molecule form ring structure. Very small tiny ring. We call that plasmid. It's very important in terms of genetic engineering. We will study that later. Prokaryotes do not have a membrane-bound organelle. No compartmentalized like eukaryotic cells. Cytoplasm bound by plasma membrane. So this is plasma membrane, this outline plasma membrane, and inside is just all cytoplasm. There is no nucleus. So inside is just called cytoplasm. On average, 100 times smaller than eukaryotic cell. There are 3 domains. One is bacteria, archaea, third one is eukaryota, but 2 out of 3 domains, such as bacteria and archaea domain, they all have prokaryotic cell. They look like this way. So single cell, and inside the cell, there's no any nucleus. Nucleoid, this is the reason DNA molecule clustered, and some small ribosome molecule. Ribosome is the site where protein synthesis takes place. Even E. coli or bacteria, they need to produce a protein. So bacteria has ribosome in the cell, also plasma membrane, this outer layer, and then cell wall, and then there's a capsule, and then there's a long tail. This is called flagella. As you see, prokaryotic cell has a very simple structure of the cell. But eukaryotic cell, some eukaryotic cell is unicellular, but most of them multicellular species. DNA located inside the nucleus. This nucleus is bounded by membranous nuclear envelope. Also, eukaryotic cell has membrane-bound organelle. The plasma membrane is a selective barrier that allows sufficient passage of oxygen, nutrient, and waste to service the volume of every cell. The general structure of a biological membrane is a double layer of phospholipid. We call that phospholipid bilayer. Living organism belonging domain Eukaryota has eukaryotic cell. So this is the structure of a eukaryotic cell. You can see outside plasma membrane, and inside the plasma membrane, the biggest organelle named nucleus. Nucleus is composed of a nuclear envelope, nucleolus, and chromatin. 3 parts form nucleus. If one is missing, we cannot say the cell has nucleus. That's the reason why we describe prokaryotic cell does not have a nucleus. Prokaryotic cell does not have a nuclear envelope. Prokaryotic cell does not have a nucleolus. Therefore, we cannot say prokaryotic cell has a nucleus. Outside the nucleus, there are several organelles is surrounded by membrane-bound system. In the cytoplasm, you can see this small dot, ribosome, and Golgi apparatus, endoplasmic reticulum, and mitochondria, lysosome, and peroxisome. Here's the lysosome, peroxisome, and you can see some long hair called flagella. Some eukaryotic cell has flagella, long hair, or sometimes some short protrusion called microvilli. Some eukaryotic cell has microvilli, not all of them. This is a plant cell. Plant cell has cell wall in addition to plasma membrane. So here's a cell wall, and here's plasma membrane, and then inside the cell you can see nucleus, ER, Golgi, and you can see central vacuole. This central vacuole is a huge space to hold water. You can see chloroplast, also mitochondria, peroxisome. These figures represent plant eukaryotic cell, which is another organelle of animal cell. Plastid. Chloroplast is one kind of plastid. Animal does not have plastid, so the answer is A. Cytoskeleton is a skeleton. Skeleton means like a bone. Cyto means some molecule act like a bone in the cytoplasm. We call that cytoskeleton. The cytoskeleton is a network of fiber extending throughout the cytoplasm. This network of fiber allows the cell maintain certain structure and shape. It organizes the cell's structure and activity, anchoring many organelles. So this cytoskeleton kind of hold each organelle in a certain spot inside the cell. Therefore, organelles have their own location. It is composed of three types of molecular structure: the microtubule, intermediate filament, and microfilament. These are the types of cytoskeleton. Microtubule is very thick microtubule of tubulin protein. Tubulin protein make up of microtubule. Intermediate filament, medium-sized intermediate filament of various protein. Various protein make up of intermediate filament. And actin protein make up microfilament. So this is plasma membrane, and you can see inside, this is the cytoplasm portion. Inside the cytoplasm, there are several organelles, and then you can see this orange line represent microtubule. This green line represent intermediate filament. You can see this purple line represent microfilament. They form network of fiber inside the cytoplasm. Therefore, they can hold anchoring organelle in certain location. Therefore, the cell overall maintains structure and shape. Microtubules are hollow rod about 25 nanometer in diameter and about 200 nanometer to 25 micron long. Function of microtubules is shaping the cell, guiding movement of organelle, and separating chromosomes during cell division. When we study cell division, microtubules come back. Centrosome and centriole, this is some other organelle, exists inside the cytoplasm. In many cells, microtubules grow out from centrosome near the nucleus. So, centrosome is the organelle to produce microtubule. The centrosome is a microtubule organizing center. In animal cell, centrosome has a pair of centrioles each with 9 triplet of microtubules arranged in a ring. So centrosome is composed of a pair of centrioles, 2 centrioles. We will see the figure here. Can you see this one? This is a centrosome. And each one, this one is centriole. This one is centriole. 2 centrioles form centrosome. Entire this organelle we call centrosome. Here you go. Here's centrosome. And centrosome has 1 centriole here, and then the other centriole maybe a little bit underneath. When you look at organelle inside the cell, most organelle has a membrane-bound system except this centrosome. Centrosome is not surrounded by membrane. And centrosome has 2 centrioles, and then each centriole is going to separate when this cell undergo division. So we will study this later, but this centrosome is the place microtubule can be synthesized. Also, this is cilia and flagella. So long hair is called flagella, and short hair is called cilia. This hair-like structure allow the cell can move. What is component of this hair-like structure? They are composed of a microtubule. Okay, so when you look at this hair, inside the hair, there's basal body. This is composed of a microtubule. Also, sperm has long tail. This tail also composed of microtubule. We call them flagella. Long hair called flagella, and short hair called cilia. Cilia and flagella are composed of microtubule. Cilia, flagella differ in their beating patterns. Cilia is just short, so beat in unison like oars, shorter and more numerous than flagella. Flagella rotate in a cross motion like a propeller. Flagella rotate in a corkscrew motion like a propeller. So the movement is more strong than cilia. Protists, for example, fungi and mushroom, they all belong to protists. They use cilia and flagella to swim through water. When sperm and egg fertilize, then they use cilia or flagella to swim. In other words, the sperm swim by using flagella, arrive to egg. So fertilization can take place. Most living organisms belonging to protists, they carry cilia or flagella in order to make a fertilization to take place. In animals, cilia usually move fluid past a surface. And female reproductive tract of a vertebrate also has cilia. Respiratory tract of most terrestrial vertebrate also has cilia, so in your respiratory tract, there is some mucus can be swept out by cilia. Most animal sperm cells are propelled by flagella. It's long hair. It's not short, so sperm has a flagella, not cilia. Now, let's look at each organelle inside the cell. First, nucleus, large organelle. House DNA. It holds DNA. Nucleus has 3 parts. One is nuclear envelope, second chromatin, third nucleolus. Nuclear envelope encloses the nucleus. It's wrapped around the nucleus. Isolate the nucleus from the rest of the cell. So nuclear envelope isolates nucleus from the cytoplasm. Double membrane is not single membrane. Nuclear envelope has inner and outer membrane, double layer of membrane. Each consists of a lipid bilayer. Nuclear envelope are studied on outer surface by ribosomes. Also, nuclear envelope continues with the membrane of endoplasmic reticulum. Since nuclear envelope is double, therefore outer nuclear membrane can extend toward the cytoplasm. It forms endoplasmic reticulum. Also, nuclear envelope perforated by nuclear pore. There is a hole on the nuclear envelope. This hole regulates the entry and exit of molecules from the nucleus. Throughout this nuclear pore, some large molecule can pass through, and then passage of a large molecule is regulated by nuclear pore complex. So nuclear pore act like a gatekeeper. This is nucleus. This is not the cell. Do not confuse that. Here, entire cell, this colorful portion represents nucleus. When you look at nucleus, you can see double layer of nuclear envelope. And you can see this pore, nuclear pore on the nuclear envelope. This yellow structure indicates nuclear pore. And also ribosome attached on nuclear envelope. And when you look at inside the nucleus, this is a nucleus, not the cell. When you look at inside the nucleus, you can see this dark portion. We call that nucleolus. Nucleolus. When you look at outside the nucleolus, inside the nucleus, you can see some string entangled. We call this chromatin. These are the DNA molecule. DNA molecules is entangled. We call that chromatin. Again, this is a nucleus. And outer nuclear envelope extend toward the cytoplasm and then folding to form endoplasmic reticulum. Endoplasmic reticulum has a dot attached on endoplasmic reticulum. This dot make endoplasmic reticulum looks rough. So we call that rough endoplasmic reticulum. You can see nuclear pore on nuclear envelope. This nuclear pore regulate passage of certain molecules. And here's inner membrane. And then inside the center, there's a dark portion. We call that nucleolus. It's not nucleus. Entire this structure we call nucleus. Inside the nucleus, there's nucleolus. And then the rest part outside the nucleolus, this string structure, we call that chromatin. This is the place where DNA macromolecule located. Chromatin, nuclear material within nucleus, in other words, DNA, composed of chromosome, DNA molecules and their associated protein. Chromatin exists with DNA, and this DNA bind with protein together. The name of a protein is histone. Histone protein bind with DNA and they located inside the nucleus. Extremely long, indistinguishable strands when a cell is not dividing. Most of the time, when the cell does not undergo cell division, the DNA is like a strand. It's an entangled strand, but this strand is composed of DNA molecule with histone protein. Chromatin condensed distinct during cell division, but this entangled strand is going to condense when the cell decides to undergo cell division. In the nucleus, DNA is organized into discrete unit called chromosome. Each chromosome is composed of a single DNA molecule associated with histone protein. The DNA and protein of a chromosome are together called chromatin. Chromatin condenses to form discrete chromosome as your cell prepares to divide. So let's arrange terminology. DNA bind with histone protein and they look like a string or a strand. We call that chromatin. This chromatin is entangled strand is going to condense to form rod structure. That condensed form of chromatin we call chromosome. So this, look at here, entangled strand, but this entangled strand has DNA and histone protein together. This state can be observed when the cell does not undergo cell division. But if this cell decides, okay, we are going to divide this cell into two cells, then this chromatin, entangled strand state of chromatin is going to condense, and then they form some kinds of rod structure, like this, clear condensed structure. We call this chromosome. So here's a chromatin, is entangled strand, but when this cell is the time to divide, then this chromatin condensed supercoiled to show this clear structure. We call this chromosome. Nucleolus, okay, don't confuse with nucleus. Nucleolus. At least one exists inside the nucleus. Nucleolus is the site of ribosome. Inside the nucleolus, ribosomal RNA can be synthesized. There are three kinds of RNA. So you will study that later, but if I introduce here first, here's messenger RNA and transfer RNA and ribosomal RNA. Messenger RNA can be produced from DNA, and this ribosomal RNA can be produced from nucleolus. Nucleolus produce ribosomal RNA. Nucleolus composed of ribosomal RNA, DNA, protein, and ribosomes. Ribosomes use the information from the DNA to make protein. I told you that ribosome is the site where protein synthesis takes place. This ribosome is composed of RNA plus protein. That ribosomal RNA can be synthesized from nucleolus inside the nucleus. One more time, here's nucleolus, here's chromatin, globalized structure, nucleus. And from nucleolus, ribosomal RNA can be synthesized. Okay, ribosomal RNA can be synthesized from nucleolus. And then here's nuclear pore. Ribosome, protein factory, small particle of ribosomal RNA and protein. So this is small, like a dot. This dot is made up of protein and ribosomal RNA bind together. Workbench for protein synthesis. Some appears as a singleton in cytoplasm, free ribosome. In the cytoplasm, this single ribosome is flowing freely. Some ribosome, they form polyribosome. They string together in cytoplasm. Some ribosome stir the membrane of the nuclear envelope and it forms rough endoplasmic reticulum. So you can see here's membrane, and then when ribosome bound to the membrane, they can be observed by transmission electron microscope. In the cytosol, this ribosome freely flowing in the cytoplasm, this dot ribosome is not attached to the membrane. Therefore, we call this free ribosome in cytosol. And then this ribosome is attached to the membrane, so we call this rough endoplasmic reticulum. One more time. Here's a cell. Here's a nucleus. Outside the nucleus is a cytoplasm. And when you look at nucleus, inside the nucleus, there's a nucleolus. Nucleolus is the site where ribosomal RNA synthesized. And then the rest part inside the nucleus, these are chromatin. You can see this yellow structure as nuclear pore. And then can you see this dot, small dot? These are ribosome. Ribosome can attach it on the nuclear pore. And then the outer nuclear membrane extend toward the cytoplasm and then folding, this we called endoplasmic reticulum. And then this endoplasmic reticulum, you can see this kind of dot. These are ribosome. Since ribosome attached to endoplasmic reticulum, we call that rough endoplasmic reticulum, RER. Some endoplasmic reticulum does not have a ribosome. Okay. Some ribosome just freely floating inside the cytoplasm. Endomembrane system, internal membranes loosely connecting compartment, segregate molecules and order biochemical processes. So the component of endomembrane system, that means inside the cytoplasm some organelle has a membrane bound system, we call that endomembrane system. For example, nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosome, vacuole, plasma membrane. These form endomembrane system. You can simply understand, all these organelles carry membrane-bound structure. These components are either continuous or connected via transfer by vesicle. Okay, let's look at figure. A vesicle is a small, tiny sac and tagged with a mailing label amino acid by rough endoplasmic reticulum that carries molecule with the help of motor protein. Bud from and fuse with endomembrane system and plasma membrane. And this vesicle participate in exocytosis and endocytosis. Exocytosis means this vesicle can fuse with the plasma membrane. Then some content inside the vesicle can be exported outside the cell. We call that exocytosis. Also, there is some cell, but some vesicle approach to the cell, and then there is some molecules inside the vesicle. When these vesicle approach to cell membrane, plasma membrane, then they are both membrane. Membrane and membrane can merge each other. Therefore, once these membrane merge into plasma membrane of the cell, then the content inside the vesicle can be discharged in the cell. We call that endocytosis, in pocketing of plasma membrane to surround material with a vesicle and import it. This is a plant cell. How do you know? Here's the cell wall, and then here's cell membrane. And another characteristic of a plant cell is plant cell has a chloroplast. Animal cells do not have a chloroplast. Another characteristic of a plant cell is this. Can you see this vacuole? Large vacuole. Large vacuole. This is the space to hold water. But when you look at this small sac-like structure, we call it vesicle. This membrane, the terminal of Golgi body, can bud off to form vesicle. But there are some molecules inside the vesicle. The vesicles can be transported. It's like a vehicle. It can go outside, it can join to other organelle. Once you join that, the content inside the vesicle can deliver to other organelle. Or the content inside the vesicle can export outside the cell. This is a nuclear envelope. So here's the nucleus. Just looking at the membrane portion of a nucleus. Here's nuclear pore. And then the nucleus body is empty in this figure. But look at nuclear envelope. 2 layers. Outer nuclear envelope extend toward the cytoplasm and fold. We call this endoplasmic reticulum. Endoplasmic reticulum. Then Some endoplasmic reticulum has ribosome attached. Ribosome molecule attached on endoplasmic reticulum. We call this rough endoplasmic reticulum, RER. Just write down only R. But some endoplasmic reticulum, there is no ribosome attached, so it looks smooth. Therefore, we call this smooth endoplasmic reticulum or SER. This is the beginning of endoplasmic reticulum, and then this side is the terminal region of endoplasmic reticulum. Then this membrane is barred off to form tiny small vesicle. Inside the tiny small vesicle, there is some molecule located. So here's endoplasmic reticulum, but this budding small vesicule can be formed here, and you can see this dot. This is rough endoplasmic reticulum. This area, there is no dot, very smooth. We call this SER, smooth endoplasmic reticulum. Endoplasmic reticulum has narrow channels forming interconnected sac and tubules. Typically make up at least 50% of total cellular membrane. You see there's so many membrane. These are all membrane. So inside the cell, 50% of total cellular membrane comes from endoplasmic reticulum. Important in the synthesis, modification, and transport of molecule. ER, we can easily call ER, has both rough ER and smooth ER depending on the presence and absence of ribosome. Function of ER. First, rough ER has ribosome attached and secrete glycoprotein. So rough ER produce protein, glycoprotein. Rough ER distribute transport vesicle. Rough ER is phospholipid bilayer. I told you 50% of a membrane comes from ER. Therefore, rough ER is the phospholipid membrane factory for the cell. SER does not produce protein. Rough ER, since rough ER has ribosome, ribosome is the site where protein can be manufactured. Therefore, rough ER secrete glycoprotein. Glycoprotein can be manufactured from rough ER because rough ER has ribosome. Since SER does not have a ribosome, protein cannot be manufactured in SER, but lipid molecule can be synthesized in smooth ER. Metabolize carbohydrate also. So from SER, lipid and carbohydrate metabolized. SER detoxifies drugs and poisons, and also SER store calcium ions. Long story short, protein can be synthesized in rough ER. Lipid and carbohydrate metabolism take place in SER. Especially lipid can be synthesized in smooth ER. Protein can be synthesized in rough ER. Carbohydrate modification can take place in SER. So if you have a clear discretion between rough ER and SER, then you will be okay. Rough ER, here's you can see ribosome, well-developed membrane. ER continues with nuclear envelope. There are two layers of nuclear envelope. Outer nuclear envelope extends toward the cytoplasm. It forms endoplasmic reticulum. Rough ER, both are ribosome stirred. So nuclear envelope also there is some ribosome attached to here, right? And also endoplasmic reticulum has ribosome attached. As membrane-bound proteins are synthesized on ribosome, some are extruded into the ER. Some proteins remain within rough ER. But many proteins are modified, properly folded, and sent to Golgi apparatus. Others, on the outside of the rough ER, function in synthesis of a new phospholipid membrane. In other words, this ribosome is the place where protein synthesis takes place. Inside the ribosome, protein can be manufactured. Those proteins, some of them just stay inside the lumen of ER, but most of them secrete to JSON organelle named Golgi apparatus. Once protein synthesized in ribosome, this protein can be modified, properly folded, then sent to Golgi apparatus. Smooth ER do not have a ribosome attached. Lax ribosome, also involved in synthesis of a new membrane. In some cells, smooth ER produce other lipids, convert glycogen, calcium ion storage, or break down harmful molecules. Just remember, SER involved in lipid and carbohydrate metabolism, rough ER involved in protein synthesis. One more time, here's a smooth ER, rough ER, and then this dot, ribosome, and this the protein synthesized inside the rough ER, and then the protein, there's some protein located inside the membrane of ER, but at the terminal region of ER, this membrane is barred off, and it forms transport vesicle. Could you solve this question? A cell's membrane is not formed properly. It is not able to act as a barrier to its environment. What may be causing this problem? Why some cells cannot have a proper membrane? Which organelle produces membrane? Think about that. So the answer is endoplasmic reticulum is malfunctioning and cannot produce the phospholipid and cholesterol for the membrane. Cholesterol is one component of a membrane. Therefore, the lack of cholesterol, that means the membrane cannot properly formed. Also, ER produce phospholipid, especially in SER, and of course RER, they produce phospholipid. If endoplasmic reticulum does not produce phospholipid, then this phospholipid and cholesterol, they are the main component for membrane. Membrane cannot properly formed. So ER is malfunctioning, then the cell cannot have proper formation of a membrane. So the answer is B. Adjacent to endoplasmic reticulum is a Golgi apparatus. Simply we call Golgi or Golgi body, flattened in a connected membranous sac called cistone. It functions modifies product of the ER, sorting, mailing labels, package material into transport vesicles. So some protein manufactured in ER, especially rough ER, this protein transport to Golgi apparatus. Golgi apparatus is like a postal office. So many kinds of protein arrive Golgi apparatus from ER, then Golgi apparatus determine, oh, this protein will go to Pasadena. This protein will go to Los Angeles. This protein will go to Whittier. Golgi apparatus sort out proteins for their final destination. To do that, they label the proteins and package proteins into transport vesicle. Shipping and receiving center. Based on structure, Golgi apparatus has a shipping center and receiving center. So this is Golgi apparatus. Golgi apparatus has shipping center and receiving center. This is a receiving center. Golgi apparatus receive protein manufactured from ER. This vesicle comes from ER. Then this vesicle hold protein manufactured from ER. Protein carrying vesicles from the ER merge with Golgi apparatus. This is membrane, phospholipid membrane, they get together and they merge, they fuse. Then the content inside the vesicle is going to discharge into Golgi apparatus. Then those proteins modified throughout inside the Golgi apparatus. The other side, this is terminal region of Golgi apparatus. We call that shipping side. This is a receiving side. The other side is a shipping side. Then vesicles carrying modified protein leave the Golgi apparatus by forming, can you see this budding? And when this membrane, terminal region of a membrane bud out, then it forms vesicle. Inside the vesicle, there is a protein, but this protein is a little bit different from the protein originated from ER. Once protein pass through Golgi body, the protein are modified, more matured. Here's receiving side of a Golgi apparatus. So protein can be received from ER, and then this side is a transphase shipping side of a Golgi apparatus. So mature protein can be shipped to for their final destination. So here's a nucleus, endoplasmic reticulum. From endoplasmic, especially rough endoplasmic reticulum, proteins are manufactured. The terminal side of ER, membrane budding out, and then it forms a vesicle. There is a protein inside the vesicle. This protein were manufactured from ER. Now this vesicle joined the Golgi apparatus, then discharge protein. Once this protein pass through Golgi apparatus, proteins are modified, matured, and at the terminal side of Golgi apparatus, another vesicle can be formed because the membrane of Golgi body can bud out, and then inside the vesicle there is a mature protein located. This vesicle can close to plasma membrane, and then the membrane of a vesicle and plasma membrane, they fuse and merge. At that time, inside the vesicle, the mature protein can discharge outside the cell. Why would a cell have a prominent ER and Golgi apparatus? The answer is C. It is a leukocyte, white blood cell, and secrete a large amount of protein antibodies into the bloodstream. In other words, you saw that just before, vesicle can be formed from ER or Golgi. So terminal side of ER, terminal portion of Golgi apparatus is going to be bud off to form vesicle. Inside the vesicle, some protein can be transported to outside the cell, or maybe some other organelle inside the cell. In case of a leukocyte, white blood cell, they fight against a foreign invader. Therefore, this vesicle carries antibody or specific protein to kill foreign invader. So it is a leukocyte and secrete large amount of protein antibodies into the bloodstream. How these antibodies can be secreted from the cell? Because protein first manufactured in ribosome and pass through ER, pass through Golgi, and then those antibodies surrounded by vesicle and secrete out to the cell. Therefore, why ER and Golgi apparatus are so prominent? Because it is leukocyte and secrete large amount of protein antibodies into the bloodstream. Lysosome, also membrane-bound sac, hydrolyze various biological molecules such as protein, fats, polysaccharide, and nucleic acid. Lysosome also membrane-bound structure, organelle, and inside the lysosome there is a digestive enzyme located. Also, inside the lysosome, pH is acidic environment. So it's a strong acid condition inside the lysosome. Food vacuoles can be formed from phagocytosis. Phagocytosis is engulfing another cell, or endocytosis, taking in molecule from outside the cell. We will see the diagram of phagocytosis and endocytosis a little bit later. Lysosome fuse with the food vacuole and digest the molecules. Also digest old or defective organelle to recycle the cell's own macromolecules and organelles. Here's nucleus, ER. From ribosome, some digestive enzymes are produced. These enzymes are packed into vesicle and travel to Golgi apparatus. In Golgi apparatus, this enzyme is modified and this enzyme is going to more mature. In other words, the functional enzyme can be formed. This enzyme also exported to lysosome. So here, this Golgi apparatus, there's vesicle formed, and inside the vesicle there's a digestive enzyme located. This digestive enzyme delivered to lysosome. So inside the lysosome, there are digestive enzymes present. Also, this cell can have some food vacuole. Outside some vesicle has some food, and then this vesicle fuse merge into plasma membrane of this cell. So food particles are taken into the cell by endocytosis. When some material enter the cell by way of a vesicle, we call that endocytosis. So here's vesicle carrying food molecule inside, and here's lysosome. Lysosome then merge with this vesicle carrying food molecule, and lysosome already carry enzyme, digestive enzyme. Lysosome can digest food molecules. A lysosome fuse with a food vacuole and the enzyme to digest the food. So you will watch bite size of animal cell.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are two types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Vacuoles. Vacuoles. Some vacuoles are temporary. Other vacuoles stay for long for the rest of the life of the cell. Some vacuoles hold food. We call that food vacuoles. Vacuole is large size of a membrane structure. So small membrane-bound structure, we call that vesicle. Vacuole is big size vacuole. Some vacuole called contractile vacuole. Many freshwater protists has contractile vacuole in their body, in the cell, and then they pump excess water out of the cell at the time that protist can move. Some vacuole named central vacuole in plant cell. And these huge vacuole hold water. Large hold organic compounds and water. Central vacuole involved in osmoregulation. Osmoregulation means water molecule in and out of the cell, and vacuole control that. Also store water. Concentration of a pigment also can be stored in vacuole, and also turgor pressure. This turgor pressure will come back when you study osmosis. So I will give explanation of a turgor pressure at that time. So central vacuole, overall, you can think that it holds water in the cell, and plant cell has vacuole. Animal cell has very little vacuole, almost none of vacuole. So here's the plant cell. See this large vacuole. This is a reservoir of water. From electron microscopic image, you can see here's chloroplast, and then here's entire cell, and here's the nucleus. But Look at the size of a vacuole, even larger than nucleus. Here's a vacuole. See, inside the vacuole, water can be stored in the vacuole. What is the purpose of a central vacuole in the cell? Water storage. Endosymbiont hypothesis. Mitochondria and chloroplasts evolved from prokaryotic bacteria roughly 1.7 million years ago. Prokaryotes gained entry into other prokaryotes without being digested and over time became interdependent and inseparable. In other words, mitochondria is independent prokaryotic cell. Also, chloroplast was independent prokaryotic cell, but they enter into other prokaryote and live inside other prokaryote and evolve together. Mitochondria are the site of cellular respiration, a metabolic process that uses oxygen to generate ATP. Chloroplasts found in plants and algae are the site of photosynthesis. Mitochondria and chloroplasts are involved in changing energy from one form to another. So, ancestor of eukaryotic cell, the host cell, there is no mitochondria, no chloroplast. But mitochondria were independent prokaryotic cell long time ago. Then these mitochondria enter the host cell. Then engulfing of oxygen using non-photosynthetic prokaryote, which becomes a mitochondrion. Once this prokaryotic cell enter the host cell, then mitochondria provide ATP to the host cell. And these host eukaryotic cells are happy about that. So they live together. And further, another independent prokaryotic cell such as chloroplast. This chloroplast engulfed to host cell, eukaryotic cell. Therefore, this chloroplast also provide energy. Therefore, mitochondria, chloroplast, they were independent prokaryotic cell. But once they enter the eukaryotic cell, such as host cell, they live together and then they provide benefit to host cell and also mitochondria, chloroplast, they are more stable and safe inside the eukaryotic host cell. So they live together. We call that endosymbiosis relationship. Mitochondria is called powerhouse, store energy from food molecule in bonds of ATP. So ATP molecule generate in mitochondria. It has this structure, two membrane, inner membrane, outer mitochondrial membrane. Outer membrane is smooth, whereas the inner mitochondrial membrane forms deep folding. We call this cristae. The mitochondrial membrane encloses two fluid-filled space. This space between inner and outer mitochondrial membrane, this space, we call that intermembrane compartment. Intermembrane space lies between the two membranes, and the center part and the matrix fills the space within the inner membrane. So the space inside the inner membrane is called matrix. Some of the reactions that break down high energy molecules occur in the fluid of the matrix. The rest are conducted by a series of enzymes attached to the membrane of the cristae. So many enzymes attached to inner mitochondrial membrane. We will go in there when you study cellular respiration. Chloroplast, site of photosynthesis, contain chlorophyll. Light-capturing pigment is called chlorophyll. Energy is used to drive synthesis of sugar from carbon dioxide and water. This is the structure of a chloroplast. Also has two layers, outer membrane, inner membrane. And inside the chloroplast, you can see some look like a pancake stacked. We call this granum, and each pancake is called thylakoid. So a thylakoid is a membranous sac stacked to form a granum. The rest, this liquid filled the rest space is called a stroma. Stroma is the space filled with internal fluid. Plastid. Chloroplast is one type of plastid. Plastid synthesize or store pigment or food molecule. So when you look at plant, of course, the leaf of plant shows green color because of chlorophyll pigment. But also plant shows different color to fruit and flower, and those pigment located in plastid. And only found in plants and photosynthetic protists hold plastid, and chloroplast is one kind of plastid. Here's plastid, and then a starch molecule can be stored in plastid. How does chloroplast and mitochondria similar to prokaryotic cell? Important. Chloroplast and mitochondria, they have their own DNA and they have ribosome. Chloroplast has its own DNA inside the chloroplast. Mitochondria also has DNA inside. Also, they have ribosome. That means they equipped to make protein by themselves. So it was independent prokaryotic cell long time ago. But once they enter the eukaryotic cell and this chloroplast or mitochondria, both sides win-win. Both are happy, both are satisfied, so they decide to live together. Okay, so we call that endosymbiosis. But what is the evidence about chloroplast and mitochondria was prokaryotic cell long time ago? Because they have their own DNA and ribosome, as like ordinary prokaryotic cell. So the answer is D. So you will watch bite size of plant cell, and this is the end of today's lecture for cell structure and function.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.